Hello friends, myself P.S. Patil from Chemistry Department. Today this is our second lecture. In that lecture we will discuss the ionization techniques involved in mass spectrometry. So in the previous lecture we discussed the basic principles of mass spectrometry. In this second lecture we will see the ionization techniques. After completion of this lecture, we must understand the following learning objectives. First, understand the basic principles behind the ionization technique. Then, second, we understand construction and working of these ionization techniques and when to use each one of them. We also understand the advantages and disadvantages of ionization technique. So these are the learning objectives. So we start with the types of ionization technique. We must know the different types of ionization techniques. So in that, the first is electron ionization, that is EI. Second, chemical ionization, CI. Third, fast atom bombardment, FAB, FAB. Fourth one, electrospray ionization, ESI. And fifth one is atmospheric pressure chemical ionization, that is APCI. And the last type of ionization technique is matrix assisted laser desorption ionization, that is MALDI. So these are the different types of ionization techniques used for the mass spectrometer. These are the references used for this lecture. So first we start with the ionization techniques in the mass spectrometry. Before starting this technique, we must know the block diagram of mass spectrometer. This is the block diagram of mass spectrometer. In this diagram, we see the first is inlet system through which samples are injected. Then it is transferred to the ionization chamber. In the ionization chamber, ions are produced. That ions are transferred to the mass analyzer and then it is collected by this ion collection system and finally they are recorded in data handling system. Okay, this is the block diagram. In this block diagram, this part, that is ion source, it is very important part through which all ionization techniques are explained. This is important part of this mass spectrometer through which all ionization techniques are explained. So for that we see the block diagram of the mass spectrometer. Then we see what is the ion source. What is the role of this ion source? This ion source convert molecules into the gaseous ion. The most common way of producing the ion involves bombarding the sample with a beam of energetic electrons. And the ion production is divided into two categories. The first category is gas phase ionization technique and the second category is desorption ionization technique. In this first gas phase ionization technique, we use a gaseous sample. It involves the electron ionization technique and the chemical ionization technique. While in desorption ionization technique, these three techniques are involved, that is fast atom bombardment, FAB, second electrospray ionization, ESI, and the last matrix assisted laser desorption, ionization, that is MALDI. So these are the two ion production categories. In that, in our syllabus, the first, that is gas phase ionization technique is involved, means electron ionization and chemical ionization is in our syllabus. So we will discuss these two techniques in the today's lecture. Okay. Electron ionization technique. We start with the first ionization technique, that is electron ionization. In shortly we call it as EI. It is the first ionization technique. It was developed in 1920s. It is also called electron bombardment ionization. Why it is called electron bombardment ionization? Because electrons are bombarded on the analyte molecule. Hence the name is electron bombardment ionization. In this technique, a gaseous sample is used. The main function of this electron ionization technique is to convert a gaseous sample into a molecular ion. In this technique, the energetic electron interact with solid or the gas phase atoms or molecules to produce the ion. This technique is called hard ionization technique. This electron ionization technique is the hard ionization technique. Why this technique is called hard ionization? Because it uses high energetic electron to produce not only the molecular ion but also the fragment ions are produced. This needs to extensive fragmentation, which can be helpful for the structure determination of unknown compound. Okay, this electron ionization is most useful for organic compound whose molecular weight is below 600, which can be vaporized easily without decomposition. Several other compounds, thermally stable or volatile compounds in solid, liquid, gas phases can be detected with the use of this technique. Okay, this is about the electron ionization. Then in the next slide we will see the construction and working of this electron ionization. This is the figure of electron ionization. In this figure we see the ionization chamber. This is the ionization chamber. Okay. Here the first is electron beam. This one is the molecular leak through which the gaseous samples are introduced. This is a repeller. What is the role of this repeller? This is repeller and this is a slit. So suppose this slit is having negative charge. So all negative charge ions are repelled by this repeller. This is the role. Okay. Then this is a tungsten filament lamp through which high energy electrons are released. This high energy electron then pass through this electron slit and bombarded on this analyte molecule. This is the analyte molecule. The electrons are bombarded on this analyte molecule. So what happens? These electrons remove one electron from this analyte molecule and we get a molecular ion. This we see in the previous lecture in the basic principles of mass spectrometry. In this case, there is a formation of molecular ion and two electrons are released. Due to this high bombardment of electron on the molecular ion, the molecule get fragmented. And the fragmented ion, positive ions are accelerate into the ion accelerating region, while the negative electrons are accelerate towards the anode, and positive ion accelerate into the ion accelerating region. This ion accelerating region consists of two slits. This is first accelerating slit and this one is second accelerating slit. This first accelerating slit is having positive charge while the second accelerating slit is having negative charge. So what happened? All the positive ions which are coming from this ionizing chamber, they are accelerate toward the second accelerating slit. So all these ions are transferred into the mass analyzer. So this is the construction and working of this electron ionization. In this chamber, ionizing chamber, 10 raised to minus 6 to 10 raised to minus 5 torr pressure is maintained. So this is about the construction and working of electron ionization. Okay. Then we see the advantages and disadvantages of this electron ionization. The first advantage is that this technique gives the molecular mass of the unknown compound, and also it gives the fragmentation pattern of the sample. The second advantage is that in this electron ionization, the large number of fragmentations are observed. Due to this large number of peaks, we get the structural information of the unknown compound. In the third advantage is it gives the reproducible mass spectra. And the last advantage of this electron ionization is it can be used as a GC or MS interface. Okay. Then we see the disadvantage of electron ionization. First disadvantage is sample must be thermally stable and volatile. Other samples are not suitable for this EI, that is electron ionization. Second disadvantage is that a small amount of sample is ionized. That means 1 in 1000 molecules. And the last disadvantage is unstable molecular ion fragments are formed so radially that are absent from the mass spectrum. Absent in the mass spectrum. In the previous lecture, we see the fragmentation pattern. In that, we see one positive and one radical forms are formed. That radical forms are not detected in the mass spectrum. Only the positive ions are detected in the mass spectrum. Hence, the disadvantage is that unstable molecular ion fragments are not detected in the mass spectrum. Okay, this is the advantages and disadvantages of electron ionization. So with this, we finish the electron ionization technique. Okay, then immediately we start the second ionization technique, that is chemical ionization. In this chemical ionization, we use a gaseous sample. This chemical ionization is a very important soft ionization technique. It is a gaseous phase ionization method. because we use a gaseous sample for this technique. In this chemical ionization technique, fragmentation is less than the electron ionization. Due to this less fragmentation, it gives intense peak of the molecular ion. We take some example. Some molecules like alcohol, ether, amine, ester, amino acid are highly fragmented in electron ionization. So, we will not get the molecular ion peak due to the more fragmentation. We not get the molecular ion peak by using electron ionization technique. To overcome this problem, we used chemical ionization technique and get proper molecular ion peak by using chemical ionization technique. Okay, this is about the chemical ionization. Then we see the steps which are involved in chemical ionization. Before that, the chemical ionization is carried out in the instrument which is similar to the electron ionization. With some modifications are there. The modifications are, the first is addition of vacuum pump, and the second is narrowing of exit slit to mass analyzer to maintain a gas pressure of about 1 torr in ionization chamber. And the third modification is that it providing a gas inlet. Okay, so these three modifications are extra in the chemical ionization instrument. The remaining instrument is similar to the electron ionization. So we see the steps which are involved in chemical ionization. These are very important. So the first step, a carrier gas or the reagent gas is introduced into the ionization source at slightly higher pressure than electron ionization. We see that in the electron ionization, the pressure is 10 raised to minus 6 to 10 raised to minus 5 torr, while in this chemical ionization, the pressure is 1 torr. So it is slightly higher, right? In this case, the carrier gas, that means methane, ammonia, isobutanes, are used as a carrier gas. So out of these three gas, in this chemical ionization technique, we take the first gas, that is methane. Okay. So this gas will ionize due to electron impact from the ionization source. The gas which is present in the ionization chamber, it reacts with the electrons which are released from the tungsten filament lamp, and they produce the molecular ion, and 2 electrons are released. So this molecular ion, due to the high electron hole, they undergoes fragmentation and gives CH3 plus ion and H radical. So these CH4 plus and CH3 plus are called primary ions. So this is the second steps of chemical ionization. In the third step, the primary ions which are formed, they will react with excess of methane gas, and it will produce a different types of secondary ion. We see that reaction. So this is the primary ion CH4 plus. It reacts with the excess of methane gas. It produces CH5 plus and CH3 radical. And this another primary ion, it reacts with the excess of methane gas. It produces C2H5 plus ion plus H2. So these two CH5 plus and C2H5 plus are called secondary ions. So in the third step there is a formation of secondary ion. In the fourth step, that secondary ion will react with the analyte molecule. In the fourth step, secondary ion react with the analyte molecule. It reacts by three ways. That three ways are as follows. The first one is proton transfer and the second one is hydride transfer and the third one is the electrophilic addition reaction. So we first see the proton transfer. Here the M, it is nothing but the analyte compound. This analyte compound react with this secondary ion CH5 plus. It forms MH plus peak in the mass spectra. So it is explained by taking one example. Suppose the molecular weight of this analyte molecule is 25. Okay, and it is react with this secondary ion. It produces peak at 26. That means one proton is transferred from this secondary ion to this analyte compound. So we get the peak at 26. Okay. So here again the another secondary ion react with the analyte molecule and we get the M plus H ion peak in the mass spectra. This proton transfer, it is generally takes place in amines and alcohols, and the ions which are produced in this spectrum are called quasi-molecular ions. These ions are called quasi-molecular ions. So this is the first proton transfer. Okay. The next one is the hydride transfer. In this hydride transfer, we see the analyte compound react with this secondary ion. It forms M-1 ion peak in the mass spectrum. That means, suppose we take the molecular weight of this analyte compound is 25, then we get the peak at 24 in the mass spectrum. That means one hydrogen get transferred from this analyte molecule to the secondary ion. So one less hydrogen is obtained. That means 24 at 24 we get the ion peak in the mass spectrum. Okay, this hydride transfer, it is generally takes place in carboxylic acids and alkanes. Okay, this is about the hydride transfer. And the last is electrophilic addition reaction. In that, this analyte compound react with the primary ion. This primary ion having a molecular weight 15. 12 plus 3, that is 15. So we get the ion peak at 40. That means suppose we consider the molecular weight of this analyte compound is 25, right? So 25 plus 15 of this primary ion. So we get 25 plus 15, that is 40. So we get the molecular ion peak at 40. Okay, this is about the electrophilic addition reaction. Then we see the advantages and disadvantages of this chemical ionization. This chemical ionization, it is used for high molecular weight compound, while in the electron ionization, it is used only for those compounds whose molecular weight is below 600. So this is the difference point between chemical ionization and electronic ionization. Then the second advantage is that it is used for sample which undergoes rapid fragmentation in AI. We see that alcohols, amines, ether, these are rapidly fragmented in electron ionization and we never get the molecular ion peak. So to overcome this problem we use this chemical ionization. So it is another difference point between this electron ionization and chemical ionization. Then we see the disadvantages of the chemical ionization. The first disadvantage is it has relatively less sensitive than the electron ionization. Second disadvantage is it is not suitable for thermally stable and non-volatile samples. Last disadvantage is that sample must be diluted with large excess of reagent gas to prevent a primary interaction between the electron and the sample molecule. Okay, so these are the disadvantages. The difference point in electron ionization and chemical ionization is in the electron ionization, pressure is 10 raised to minus 6 to 10 raised to minus 5 torr is maintained, while in the chemical ionization, 1 torr pressure is maintained. So this is the one of the difference point between chemical ionization and electron ionization. Okay, so student, you write down the difference between electron ionization and chemical ionization by studying these two techniques. Okay. So in the next lecture, we see the instrumentation of mass spectrometry. Okay. Thank you.